Good morning, and we're back here with the king of rock and roll, explaining how meiosis results in the transmission of chromosomes from one generation to the next, right? So this guy's got 46 chromosomes in each cell, right? So does this lovely lady, Priscilla, right? But they are going to do this, some of them, they're going to do this cell division, which produces cells with half of that number of chromosomes, which are on their way to this lovely lady, right, Lisa Marie. And when she gets 23 from each of her two parents, right, she again has 46. But these chromosomes have been transmitted to this wonderful generation. Now the details. So here we have the idea in a kind of a diagram, very general kind of diagram, right, that says all that stuff, right? We have the words haploid and diploid. Remember what they mean? Haploid means not half, right? HA for half, HA for haploid, not a hard way to remember it, not a bad way to remember it, but it means one set, right? What does set mean? One of each kind, which in a cell that has two sets, a diploid cell, right? There are two of each kind, right? And so we have that one of each kind idea. The pairs of similar ones called homologous, right? That word's going to show up in a few different places, okay? This is part of sexual, not asexual reproduction, right? We're going to produce diploid organisms by putting together two haploid gametes. So what does meiosis result in? Cells that have half the number of chromosomes, but more importantly, a very specific half, one of each kind, one of each pair, one of each size. Remember, we said that in a whole bunch of different ways, right? So now here, there's all kinds of steps, things that you could call steps or stages that occur in a sequence in meiosis, just like mitosis. Here they don't ever say anything about knowing those steps specifically, but they do say they want you to know that this division occurs in two steps, stages, however you want to say that. So we'll keep an eye on that, right? And here they tell you that something we might have mentioned before, sporophyte, gametophyte, there's different ways that have evolved in terms of a reproduction cycle, right? They're not going to ask you anything about any different kinds of cycles than the general animal model, which you show right here, since we are animals. Okay, so now we're going to go to meiosis 1, right? Because that's the place where the separation of these pairs happens, and that is what ensures that each gamete is just going to get one set, right? One of each kind. So that would be here, right? Here's a cell with two of that kind, of which there's only one kind in this cell, right? There's just one kind of chromosome, two of them homologous pairs, and this meiosis 1 has accomplished separating and producing cells with just one of each kind, one set. Diploid here, right? Haploid here, and then meiosis 2 accomplishes separating the copies. Copies, where did they come from? Do not forget that meiosis, just like mitosis, is preceded by an interphase during which there is a G1, an S phase, and a G2, but most importantly here an S phase, where single copies of DNA get replicated and those copies are momentarily stuck together in double-stranded chromosomes. So now we have the idea of how does this generate genetic diversity, which is a key idea in evolution. And since they took evolution out of this, they're not going to have that implication of this diversity in an evolutionary sense, but still the creation of it, without a doubt. You're going to see this on the test in some way, shape, or form. So they identify for you how the process of meiosis generates genetic diversity, and that's two very specific direct ways involving those processes called crossing over and random or independent assortment. Remember, this is a word that Mendel used. That's the next video. Independent assortment. Here they just call it random assortment. They should really emphasize that word random for all of these processes, crossing over, the thing they call assortment, and then the subsequent fertilization. They really here should note that that is also random. All three of those processes increase variation. Two of them, the crossing over and the random assortment, happen directly in meiosis, and then the gametes produced by meiosis are going to take part in that third fertilization part. Okay, so first, the random assortment. What does that mean? It means that these chromosomes, which here have been shown to be blue and red for you, showing that the blue ones are what came from the dad of the organism that whose cell they're featuring here, and the red ones originally came from mom, right? Maternal and paternal, that in relation to each other, they can line up either this way or that way, and that can produce gametes that have the two blue ones, the two red ones, one blue and one red, or one blue and one red the other way, with the big ones and the little ones alternating. And if you are an organism that has 23 such pairs of chromosomes, then you have the ability to make, just for this reason, some, I forget if it's 6 or 8 billion, a bunch, right, of different gametes just for this independent assortment reason. Now we tack on to that the fact that crossing over happening early on during prophase 1, right, which we don't need that name, but you bump into it. That process whereby these homologs come together, called synapsis, but that didn't make the blue boxes, right? That this process of crossing over, however, does happen during meiosis 1. They want you to know that part, not specifically prophase 1 part, but that. So don't forget that. It's in the blue box, right? What does this involve? Homologous chromosomes are going to have segments break off and switch places, right, between each other. Clearly illustrated here with the red and the blue, very helpful for that. And so we end up here, as you can see, with chromosomes that are part of what used to be the paternal one, part of what is now the maternal one. You can see that very clearly in these diagrams. And they're going to be separated, right? These homologous chromosomes. When are they going to be separated? Well, specifically in anaphase, but what they want you to remember for sure is that we are still in meiosis 1. When this separation, that makes sure that the gamete is going to end up with one set of chromosomes, that happens in meiosis 1. Okay? Also in meiosis 1, again, they line up in different possible ways, independent assortment, and the crossing over happens. So now in metaphase 2, right, these sister chromatids are separated, and since they want you to know about this, and it really takes both of those separations in meiosis 1 and meiosis 2 to produce a eventual cell. This will eventually be one cell, that'll be another, right, that has just single-stranded copies of one from each pair, one set. We really need to add anaphase 2 and meiosis 2 in this story of how we get to one set. So if we now just emphasize again this list of 1, 2, 3 ways, 1, 2, 3, independent assortment, crossing over, and random fertilization. If we add to the list of genetic diversity causers, the thing that in the next unit we'll get to a lot, mutations, they hold a special place, remember, because they are the things that ultimately create different kinds of genes, alleles, that might be crossed over, because if you switch two things that are identical, you haven't really changed anything. So mutations are in this list, but here we're focusing on the variety causers that have to do with meiosis and then fertilization. Therefore, again, sexual reproduction involves these and asexual doesn't, just to emphasize that. Okay, so now we get a short one. We're bringing mitosis back into play because they want you to describe similarities and differences, right? Here's the only time they say phases, just kind of threatening that we might talk about metaphase 1 and metaphase of mitosis, but I don't think they will because they didn't say that specifically, but they do use the word phase here. All right, now here I would take exception to the way they worded this is very misleading. So pay attention. Here we go. Mitosis and meiosis are similar in the fact that chromosomes separate. There's some kind of separation, and Mendel used the word segregate, and they chose to use that word here. But the way that they segregate is quite different when you're talking about meiosis 1, where homologous pairs separate. That kind of separation never occurs in mitosis, so they really should have, I'll have to make a phone call. They really should have worded that part differently, right? But they're emphasizing that in both, a similarity in both, there is some kind of separation of chromosomes, but how they differ is in the number of cells produced and the genetic content. The genetic content in two different ways, that they have only half the number, right? That much content. In meiosis, the resulting cells have half the number, but also in the fact that the content is different in every other cell, which we emphasize right here, right? Mitosis produces 2 as opposed to 4. That's a content thing. And identical genetically as opposed to different genetically, that's another content thing. The similarity, it comes in the separation again. And even though they didn't make the diagram here in meiosis, remember that this separation of sister chromatids is exactly the same as it is in meiosis 2 as it is in mitosis. So we're over and out. Hope you have a wonderful day. Isn't that a cute kitty? Or two kitties?